Yeah, I think I'm actually working on recording, so we will get started. Okay, mm, we're going to start off with a review problem. Oh, give me 1 second. We're going to start off. We're going to start off with a review problem. I'll give you guys like 3 minutes. These are three questions. They should be pretty straightforward. They're from the 2011 open exam. Yeah, and then afterwards we can talk about the questions. That's it. Yeah, yeah. I have the questions up at 7:10. We'll discuss what the answers of the question. Once you guys have your answers, or if you guys want to go like question by question, or you just want to unmute, you guys can say your answers either in chat or unmute. Okay, let's talk about it. No. Okay, let's talk about it. If anyone has any final answers, put them in the chat. Otherwise, let's discuss. So for number one, from both the answers I've seen, I got A, sorry, E. Um, for number two, C, and for number three, I got one C and one E. So number one and two are correct. Um, and just to quickly go through it, all of the things require hydrogen bonding. If you're forming an enzyme, if you're forming any proteins, it's going to be its secondary structure. Um, ice crystals also require it. That's the crystalline matter structure. And DNA replication, the A and the G, or sorry, A and T are bonded together with two hydrogen bonds, and C and G are bonded with three, or vice versa. So, um, and then for number two, lowest pH, of course, is lysosome. It's the one that's digesting everything and breaking it down. So it's going to be very acidic. It's going to have enzymes. It's going to have low pH, and that is why it's dangerous if the lysosomes open up in your cell because they're all going to cause the pH of the cell to be too high and cause everything to denature. And then for number three, I got statements two, three are true, or all the statements are true. So C is the correct answer. And let's talk about why. So we know that two and three are true, at least everyone's agreeing with that. But oh, C, D, two, three. So it says that lipids, for number one, it says lipids are made up of polymers of fatty acids. So, um, in lipids, it's kind of a different story because there's no monomers per se of fatty acids. Like and lipids could be different amounts of fatty acids. Like a typical lipid, like a phospholipid could be three fatty acid tails, and then you can have other stuff in it besides fatty acids, other components. So the monomers and polymers concept is not very applicable towards lipids, and that's why it's going to be C thing two and three. Okay, but anyways, let's go on to the main point of this. So we're going to talk about ATP and coupling reactions. So it's going to be about cellular reactions, and afterwards, going to go back and reinforce some of the um earlier stuff we learned. Okay, but anyways, So, there is something. So, when you have ATP, you have reactions that are happening simultaneously in two parts of the cell. So, energy coupling is the use of using an exergonic process to drive an endergonic one. For example, when you break down ATP, you're releasing energy. It's an exergonic reaction. However, if you create a protein, that's an endergonic reaction because it requires energy to um, like it requires energy to maintain a more stable structure, like where if you're making like cellulose or something. Um, and so ATP drives this process and it's kind of the cell currency. So let's break down what ATP is. ATP is a sugar, it's a ribose sugar. So you got ribose. And then attached to it is going to be a nitrogenous base called adenine. We'll just make this for A. And then there's going to be three phosphate groups, T, T, T. And this is also the building block for RNA, ribonucleic acid. It's the adenine version, adenine nucleotide. So when ATP is hydrolyzed, the phosphate bond forms it like makes it forms with the hydroxide group forming an inorganic phosphate. So you'll have like HOP um and that is also going to leave the other side with an inorganic, with ADP, adenosine diphosphate, because there's going to be two phosphates since this is broken apart like over here. Um, it is an exergonic reaction and it releases 7.3 kilocalories of energy per mole of ATP hydrolyzed. So every single time you use 1 mole of ATP in the reaction to break it down, you produce 1 mole of ATP, mole of inorganic phosphates, and 7.3 kilocalories. However, that's only in standard conditions. It's really important because your body is not at standard conditions. Your body is at a higher temperature, and in a normal cell, you can expect that instead of 7.3 kilocalories being released, it's actually 13 kilocalories. And that is a 78% difference because it's not at the same standard conditions. And hydrolysis releases energy, but none of the bonds are high energy. So some people will make a reference saying that the third phosphate group is a high energy bond. There's no such thing as a high energy bond. It's just because of um because of repulsion, it doesn't want to bind together, and so you're actually releasing energy when you break the bond and add the water to it, when you hydrolyze it. Um, and again, from last time, breaking the bonds don't make energy, but producing the bonds do, and so you'll see a net release of energy. And that's because one thing, like if we go back to the first lesson, we talked about how phosphate groups are negatively charged, and that's really important because if all three of these are negatively charged, say you have three negatively charged magnets, like three magnets for one side, they don't want to be next to each other, so they're going to repel each other. And if you had them bonded together and they're constantly trying to repel each other, their bonds are not very stabilized. And so taking this third phosphate group off is very easy. It's like a spring waiting to be released. And when you have a spring, it flies out. There's energy that's being expended in order for that to happen. And the same thing's happening with the phosphate. Um, Now, you can use it for cellular energy. And so when ATP is hydrolyzed, it releases energy and it heats the surroundings. So, for example, if you're breaking down ATP inside water, it's going to make the water warmer. But heat isn't really the best use for ATP because ATP's energy could be harnessed in other ways that are useful for the cell. For example, it performs things like chemical transport and mechanical cell work. And so when you're pairing it with something else, what's going to happen is you're going to create a phosphorylated intermediate molecule. And so here's what happens. You have your ATP. We're just going to assume this is the three phosphates, and you have your molecule. Your phosphate is going to break off and it's going to attach to this molecule and it's going to be a covalent bond um to to like to make a conformational change. And so that's that instead of having just the phosphate being released and the energy being released to the surroundings, you're gonna have the energy added onto the molecule because you're or like at that stage because you're absorbing energy when you create a bond. And so that's how you're able to use the energy from ATP for the molecule for your purpose. Um, but what also happens is that this molecule, once a phosphate is added onto it, it's going to be called a phosphorylated intermediate, and it's going to be super unstable because it has an extra phosphate group that it doesn't want. Its bonds are going to slowly shift a little bit because it just created a new chemical bond, and it's going to want to break apart. And so that's how you're going to be using that energy, incorporating it into breaking the bonds in the first place. Now, for um, this can be used for a chemical reaction. That's how you pair it with a chemical reaction. And for transport or mechanical work, ATP hydrolysis often leads to a change in the protein's shape and ability to bind to another molecule. So basically, if you have this transport protein inside your cell and it's right now closed, adding a phosphate group to this protein will cause a conformational change. Now, in certain proteins, and what's like targeting, what's wanted is that protein change is going to make the channel much more receptive for things to go through, materials to go inside. And for mechanical, it's basically for your motor proteins, as we talked about before. It's when phosphate gets added, it's basically inching closer, and so every single movement is going to be using an ATP. And all of this is done through a cycle because the use of ATP is exergonic and the regeneration of ATP is endergonic. You're constantly using millions of ATP every single day. Fun fact, if you don't regenerate it, you'll be expending your body's weight worth of ATP every single day. And that's unfeasible because how are you supposed to recreate that? And so what they do is you can regenerate ATP by adding the ATP and phosphate group together through other creation of energy. So it uses external energy sources to power this regeneration. Like, for example, animals use cellular respiration. Plants also use cellular respiration, but they also use sunlight for like photosynthesis, create the sugars, and they also use it to um, put the phosphates out. And then you're going to have enzymes. So through all these chemical reactions, you're going to see that sometimes it doesn't happen immediately. You can have a spontaneous reaction that is likely going to happen in the universe, but it's going to occur at a very slow pace. Because just because something might be spontaneous or more spontaneous doesn't mean that it's going to happen fast or immediate. And sometimes these speeds are so insignificant that speeds are so slow that it's basically insignificant. But if you add an enzyme, specifically a catalyst, it's gonna make the reaction go much faster. So an enzyme is a macromolecule, and actually a catalyst, and it's a chemical agent that speeds up a reaction without being consumed by the reaction. So you're going to have your enzyme that is going to be the exact same as it was before the reaction, and it can be reused multiple times until it starts wearing down. And it's really important for keeping things moving in a metabolic pathway, because if you have one reaction that takes, let's say hypothetically, an hour, and you have another reaction that takes a second, you're going to have slowdown, you're going to have buildup, and that reaction that takes an hour. Now, the reason why some of these things are really slow to happen is because of the activation energy burden. So the activation energy barrier is essentially saying in order for something to break apart, you need to make it unstable in the first place. And so with that, you need to put in energy in order to make a bond stretched out, make it break so that it can go into its smaller version. Um and this awkward state that I was just talking about of the bonds being stretched, being bended, is called the transition state. And it's when the molecules have absorbed, it's when the molecules have absorbed enough energy for the bonds to break, and it's quite an unstable molecule. It's at the highest amount of energy that the molecule can have. So you'll see graphs that go like this, and this is going to be your transition state. Because this is how much energy you're spending, this is how much energy that's not being released, and this is your final reaction. And so you are putting in energy to this molecule to make it much more stretched, much more bended, and then afterwards you can break it apart, release a bunch of energy into the surroundings, and have your smaller molecule. And so this part over here is the activation energy, and it's denoted with an E under case subscript A. And so let's specifically talk about what is happening with your enzyme and with your substrate. So a reactant will bind to an enzyme, and that reactant is going to be called the substrate. And there can be multiple substrates inside one reaction and binding to one enzyme. And so the enzyme substrate complex is a combination of all the enzymes and substrates. And so the reaction is the reaction is catalyzed and it's very specific to what it is. It's like a lock and key mechanism with a pocket or a groove that consists of like it has a small segment of its R group amino acids that are unique to a specific compound. So you could have your entire protein, and the only thing that will matter are these amino acids right here. This will be your groove, and this is where your substrate will go into to bind. And so that specific amino acid sequence is the most important. But the reason why we have all the other amino acids surrounding it is because we need a certain shape for the enzyme. Now, this sequence is going to be called the active site. That's what it's going to be referred to. And it's a binding site where everything reacts to. And as a substrate enters the active site, the enzyme is going to change shapes. Because remember, when you form bonds, you are changing the protein structure. You're causing a structural change or a structural morph that makes it into a different reaction. I mean, a different like protein in different configuration because of different chemical groups like weak bonds, like hydrogen bonds, and it makes oftentimes it'll make something that's more snug. It'll make something called induced fit. And so let's take an example of keeping your hand out for a handshake. You're gonna have your hand out like this. It's gonna be straight, it's gonna be flat, and you're waiting for someone to put their hand right next to you. But once that happens, you're gonna grip your hand closer together and you're gonna make it so that it's wrapping around sorry. You're gonna make it that it's wrapping around that person's hand. That's what induced fit is, and that happens after you have that chemical reaction. Okay, I believe that's better now. Okay. Okay, and so let's talk about the enzyme shape and why it's important and its function. So, the enzyme shape causes it to have, causes it to be held, like the enzyme is held together by weak interactions such as hydrogen and ionic bonds, and the R groups of amino acids are the ones that are forming the active site. So it has to undergo a change before it can bind to the next enzyme, and they work really well because of their shape, and so very few of them are actually needed. And the local environment could affect the shape, could affect how the protein is working. And so up to a certain point, the rate of enzyme reaction increases with temperature increasing. But beyond that point, it drops sharply because these interactions are disrupted, the shape is disrupted, there's protein denaturation, and there's an optimal temperature that's going to be given per each heat change. PH is the same exact thing. When you have more photons, when you have a more acidic environment, or when you have a higher temperature, these enzymes could cause these bonds could break down. They could have different weirded proteins, and it doesn't allow for the enzyme to bind correctly. Now, enzymes are really important because they're the active site. Because the active site provides a template which the substrate can be in proper orientation for the reaction. Because if you are having two substrates that need to bind together, you need to have them in the correct reaction site for the reaction to happen in the first place. And also, it will stretch the molecules. So we were talking about the intermediate stage, and when you bind to it and when you have that active site, it can stretch the molecules towards this transition-like form, stretching and bending critical chemical bonds that need to be broken. And the activation energy is proportional to the difficulty of breaking bonds. So when you stretch these things, when you make them distorted, you're making it easier to break down. Also, each protein has R groups which are either acidic, polar, non-polar, and so those create a microenvironment for the enzymes with either really low pH, with an acidic R group, or with the right polarity. And also, sometimes it's quite rare, you'll have direct participation of the active site with the substrate by having a brief covalent bond between the two. And this will be probably between the side amino acid chain of the enzyme, and it will contribute to the reaction. But afterwards, there will be measures to reverse it. Now, when an enzyme is saturated, the rate of reaction is determined based on the speed at which the active site converts substrate to product and not how much substrate there is. So if you see over here, uh, if you see over here, when you have no enzymes, the reaction is going pretty normally, very slowly. Okay? Over here, you see that the rate is capped. It's not going all the way up like this. That's because at these stages, it's saturated. All the enzymes are being used. So what you need to do is you need to add more enzymes. So this has the most amount of enzymes, this second most, and this is not. So that's why when you have the most amount of enzymes, it happens faster because you're not saturating all of your enzymes as soon. When you have less enzymes, you're saturating it sooner, and so less of the reaction can take place, and you have more time in order to reach this maximum stage. So next we're going to talk about cofactors really quick. There are many enzymes which require non-protein helpers for catalytic activity and may be bound tightly to help the enzyme. If it's an organic molecule, which means it has carbon and hydrogens, it's going to be a coenzyme. And oftentimes you'll have essential vitamins, you'll have elements like transitional metals that are being used as coenzymes or cofactors for chemical reactions. They're going to be really important because if you don't have that, the chemical reaction can't take place in the first place. And then finally, we have the enzyme inhibitors. So when an inhibitor binds through a covalent bond, it's typically irreversible. So as we talked about in our first lesson, there are different stages of bonds. Your van der Waals bonds are the lowest. Your hydrogen are a little bit stronger. Your ionic are a little bit stronger than that. And then your covalent bonds are the strong ones. So when your inhibitor, when you have an enzyme and you have an inhibitor molecule, and this is a covalent bond, it's pretty much glued together. It's going to be really hard for that to come off, and it's going to cause the enzyme to be occupied for good. And so a lot of toxins and poisons have this. An example is sarin gas. It's a type of nerve gas, and it has small molecules that bind to the R group of serine. So there's a specific amino acid, as we talked about earlier, called serine, and that R group and this gas binds to it and causes it so that nothing else can bind to it. And so when you have that, your the reaction that needs to take place with this enzyme can't happen in the first place. But then there are also temporary inhibition. And that's when these are weak bonds. So this could be, let's erase this, let's try this again. If this is your inhibitor, there are weak bonds like van der Waals, hydrogen. That's all reversible. And so when you have something like this, it's called temporary inhibition. And what happens is now we're going to work with concentration. Because here's the thing, if you have a permanent inhibition, once that molecule binds to the active site, it's done. It can't be reversed. You can't um you can't like take it back and it's gonna permanently be there. However, in temporary inhibition, you're gonna have it for a short amount of time and it's gonna be constantly switching. So let's say this. You have a high concentration of competitive inhibitors. More likely than not, at any given moment, your enzymes are going to be occupied by the competitive inhibitors temporarily. However, because it's temporary inhibition, if you increase the concentration of substrate you have, then your enzyme is going to be more likely occupied by your actual substrate instead of the competitive inhibitor. And so for that, it's based off of kind of like the spontaneity thing we were talking about last lesson, and it's about what's most likely going to happen and the probability that's going to be able to bind to it. On the other hand, non-competitive inhibitors work a tiny bit differently because in non-competitive inhibitors, they're not fighting for the same spot. So, for example, um, say this is your active site. This is your active site. If you have something that binds to this and causes a conformational change, this active site won't like accept these things anymore. So then what happens if it's not accepting anything? It it's going to stop the enzyme from working, and it doesn't matter how much of the active substrate you have, it depends on the concentration of the non-competitive inhibitor you have. And so they work a little bit differently and they block a little bit differently because now it's not in the control of how much of the substrate you have. And so you can see that kind of happening in this graph. When you have normal enzymes, your enzyme works like this. It's pretty normal. The reaction takes place. Um, and as your substrate concentration increases, your rate of reaction increases. So basically, if you have a lot of substrate over here, your rate of reaction is going to be really high. On the other hand, competitive inhibition is a little bit lower because think about it. If you have like a mid-level number of if you have like a mid-level um of substrate concentration, it's more likely that your active sites are going to be bound by uh, like competitive inhibitors. You won't have active sites that bind to the actual substrate, and so therefore the rate of reaction is going to be lower. However, if your substrate concentration is really high, like over here, it's insignificant because no matter how much of a competitive inhibitor you have, the rate of reaction is still going to be really high because of the concentration and the probability that it's going to be occupying its place. And then you have the final one of non-competitive inhibitor. And you realize that it levels off much earlier, around halfway. And that's because, again, they're not fighting for the same spot. They're not fighting for the same site. So the concentration doesn't matter. So long as the non-competitive inhibitor is going to be binding to the other side of the thing, it doesn't matter how much substrate you have, as it's just going to be blocked permanently. And in your permanent inhibition, do something similar to non-competitive inhibitors. You'll have some, but it's gonna be pretty stagnant. Okay, I think that's it. Okay, that should be it. So now we're gonna cover um we're gonna go a little more in review and in depth into what we did before because whoopsies. A lot of the things that we have done have been very surface level. But what we need to do is go more in depth into specifics for the Useba. So when time comes for examples, you're not lost in what they're asking or the terminology. So we're going to start off with eukaryotic cells. I'm going to start off with organelles. Okay, and we are going to start off with endoplasmic reticulums. And I'm gonna be asking questions. If you guys can just like um speed put it in the chat, whatever you remember from last time when the question comes up, that would be perfect. So let's start off with drawing or visualizing what the ER is. So what is the RER enclosed by? Yes, bilayer. Okay, good. Um, so your RER is enclosed by a bilayer, and it is going to be an extension of the nuclear envelope. So as we talked about before, you have your nucleus, and then extending on from that nucleus is going to be your rough endoplasmic reticulum. Now, something you need to keep in mind is that this endoplasmic reticulum or rough endoplasmic reticulum is going to be continuous with the nuclear envelope. Um, and so if anyone remembers, what are these tubes and sacs of the ear called? So actually really close. It's called the cisternae. Um, it's like a few letters off. Cis. And so it's kind of like the pancakes of the pancakes of the ER. Now, the cristae, which you were talking about, is the pancakes of the mitochondria. It's the folds of the mitochondria inside that encloses the matrix, the mitochondrial matrix. So they're a little bit off in the word, they're a little bit different in the wording, but they essentially the same thing just for different organelles. One is for the ER, which is the cisternae, and it's about talking about the folds and the sacs. And so now what is this inside called of the ER? Okay, I'm gonna give it away because we low-key need to rush a little. It's called the ER lumen. So now you have your basic parts, and so you have your lumen inside the ER, you have your cisternae, which are the stacks inside the ER, and then you have your actual phospholipid membrane, which is continuous with the nuclear envelope, and that's the endomembrane system. So then, what is the difference between rough ER and ER? The difference is that there's ribosomes on the rough ER that are on the surface of the membrane. So let's draw little ribosomes. And so the whole point of ribosomes, as we talked about before, is for secretory proteins and proteins on the cell membrane. Because remember, sometimes you want to have transport proteins in the cell membrane. What do you need to have? You need to have protein that is embedded inside the cellular membrane, and that needs to be created by the rough ER and stuck inside the middle with non-polar amino acids inside the center and polar amino acids on the sides, because you have a hydrophobic and hydrophilic regions. Um, these proteins accumulate in the ER lumen, where they fold and acquire the 3D shape. Remember how we talked about in the rough ER, there are enzymes that are constantly changing, adjusting, and making post translation protein modifications to the ER. So let's write that down over here. Post translation modifications. Sorry if you guys have a little trouble reading my handwriting. Anyways, so, what does these include? These includes like hormones like insulin, glucagon, growth hormone, prolactin. They include glycoproteins, which are, like we talked about before, glucose on top of carbohydrate on top of protein. These include antibodies. These include hormones like a follicle stimulating hormone, luteinizing hormone. They include digestive enzymes like pepsin, trypsin, anything in your stomach, pancreas, small intestine, and they also include your major histone compatibility complex, MHC, which is what your T cells use. You talk about this in COVID. There's a huge talk about MHC2, and that's really important because your T cells are the ones interacting with this, and this is right inside the cell membrane. And so the RER lumen is for protein modifications, and there's one specific protein modifications you should know about, and that's glycolysation. So let's bring this over here. Main one is glyco. Oh my. And so basically what's happening is that oligosaccharides are being added to the proteins. Okay, so there's words called oligosaccharides. I don't know if I talked about this before, but it's basically like polysaccharides, monosaccharides. It's just additions on top of your proteins. We talked about this when we were talking about glycoproteins, how there are things that are added on top of proteins in order to make it a glycoprotein. Those are your oligosaccharides. Um, and so that's what the ER lumen enzymes do. And there are a few modifications that you should especially know about. And so the first one is N-glycosylation. So, this is a very specific modification that regulates the polypeptide folding inside in the lumen. So basically what's happening is there's an amino acid called asparagine. So I really hope you guys looked at Campbell, looked at or at least looked at amino acid chart, like I said in the first lesson, because here's where things are gonna start hitting on. You need to know your R groups and you need to know where things are. So asparagine has a nitrogen inside of it. That is why you have the N over here. And basically what happens is that it's connecting this nitrogen inside on top of this protein, and that's why it's called the N-glycosylation, and it happens in a lot of proteins. This is a very specific oligosaccharide, and it's the thing that's binding to this protein to make the modification. Now, what happens is when you add a carbohydrate, you're changing the orientation of the protein, and you're causing it to make a specific shape. Then there's another one called O glycosylation. Glycosylation, and this is going to be with serine. slash three. So basically, what happens is this: these two amino acids, serine and threonine, have oxygen atoms inside of it. Um, and what happens is basically you're binding the protein, sorry, you're binding the oligosaccharide with this specific oxygen atom, and it's gonna create it's gonna create like this it's gonna create like this protein modification that's gonna cause a specific folding. These are two post-translational modifications you should know about glycosylation because they can talk to you about it in specific proteins or specific examples, especially in the semifinal exam. And then you have your then you have um, rough yard, which is abundant in specific places. Now, the whole concept of rough ER is you're trying to create something inside, you're trying to like create a lot of proteins. And so sometimes you're going to need a lot of proteins in some excitatory or inhibitory or in neurons, specifically neurons that create peptide neurotransmitters. Now, we're going to talk about this in the nervous system chapter. However, there are groups of neurotransmitters, and some of them are protein based. They use proteins, I think norepinephrine. Um, I don't know off the top of my head. We'll talk about this over there. But there's a specific group of RER. There's specific bundles, and they're called Nissl bodies. And basically, Nissl bodies are large granule bodies with RER. And their whole purpose is to synthesize peptide neurotransmitters. And these Nissl bodies, they'll stain basophilic. So basically, as we talked about last chapter, I believe, or sorry, last class or maybe two classes ago, you have staining, you have fluorescent staining, you have these dyeing and imaging, and so they're going to be staining dark blue and purple with a basic dye because they are so concentrated. And this happens because of their acidity. So the RER has a lot of rRNA. That's a specific ribosomal RNA that is produced in the nucleosome. Sorry, nucleolus, and that is very acidic, and so that's why there's a dark pigmentation that is produced underneath staining or microscope. And then let's talk about your smooth ER. So we mentioned before, it doesn't have any ribosomes on its side, so it's smooth. And the main purpose of the smooth ER is for steroid hormones, or creating lipids or creating cholesterol. Um, and so SER is quite abundant in your gonads, like testes, your ovaries, and the corpus luteum, which produces estrogen, progesterone, testosterone. And it's also important in your adrenal cortex where you have a lot of glucocorticoids like cortisol and um, aldosterone. And one specific SER, which is really important, is wait, let me write this down. Um, and so you have a specific RER called sarcoplasmic reticulum, like we talked about um a few classes ago, and they are common in muscle cells along with cardiomyocytes. which are your heart cells. And as we talked about before, they are storing your calcium ions. So they're releasing the cytoplasm to monitor for muscle contractions, and they basically uptake their large storage unit and their pump for calcium ions because it's going to help stimulate it. Because remember what we talked about last time. You're going to want to create these chemical gradients. You're going to be wanting to create this um like this high concentration in one end and low concentration in another end. So when you release all of this, it's going to create like this stimulation inside the cell. And so the SER is also very important in detoxing and cleaning out your system. And so it's going to be quite important in the liver because all of your blood goes to the livers before it gets traveled throughout the entire body because you don't want anything toxic going into your blood. A lot of alcohol is being transferred through the urine, and basically it's going through the hepatic portal thing. And the most common type of detox in a smoothie R is going to be hydroxylation. So basically, what's happening is that an OH group, a hydroxide group, is being added, and what this does is it makes it more hydrophilic. So what can happen is if you have something that's hydrophilic, it's water loving, right? So it can dissolve in water. It goes into your kidneys, goes into your glomerulus, gets excreted in urine. So that's how you're going to be able to get rid of all these toxic substances from your body. So what happens is you're adding a hydroxide group, it's becoming soluble, it can dissolve in water, it can go into the kidneys, it can be secreted through the urine, and you will no longer have that toxic substances in your body. That is also why when you have increased alcohol equals to more SCR, which equals to tolerance, which equals to increased dosage, which goes back to the start. Uh, um, and that's how you get this dependence. Um, and your hydroxy group is going to be added by you don't really need to know this. It'll be nice to know, but you don't really need to know it. It's gonna be added by the cytochrome T450 hydroxylase. But then again, sometimes this process can fail. Um, Actually, you know what? Here's what we're gonna do. We can Okay, the call ends in 3 minutes. Um, we can end right there for now. And then next time when we meet, we will continue working on continuing working with ATP. Um, the talk about endodermal versus exodermal processes like cell respiration and photosynthesis. And then in the second half of the lesson, we can continue with the ER and then move on to the Golgi apparatus. Is that fine with everyone? Okay. Well, if it's fine with everyone, then I think we can end it for today. Um, if anyone has any questions, you guys can ask. I know I went quite far, sorry, fast, but yeah.